What if you could eat and never be overweight, or get perfect grades on all your exams, or see woolly mammoths roam the earth again? Or what if, as the mammoth returns from the Ice Age, Down syndrome or sickle cell anemia instead could become extinct? What if the thousands of disorders and diseases that affect humanity could be fixed even before they occur? Now, thanks to an emerging gene editing technology, patients actually have the chance to fix these genes and avoid giving them to their descendants. Again, does this sound too good to be true? Until the epical work of these three visionary scientists, it was, but now it is just a matter of time.
After identifying these genetic mutations, one thought is, why don't we go into the cells and repair these mutations?
French microbiologist Emmanuel Charpentier, American structural biologist Jennifer Doudna, and Chinese-American bioengineer Feng Zhang were awarded the 2016 Tangprize for their development of the revolutionary gene editing platform CRISPR-Cas9.
Our Cas9 system is a very effective genome engineering technology that is used in a programmable fashion and allows to act on genes in a precise way to either delete genes or replace genes.
As an analogy, we can think of our genomic DNA as a necklace made of 3 billion pearls, where each pearl represents a base pair in the genome. If one of the pearls has a hairline crack, it would be difficult to find and replace it with so many pearls. But recently, scientists have found a new genetic tool called Cas9, which is equipped with its own GPS. This GPS can be programmed simply by entering the genetic coordinates. The scissors are then guided to the cracked pearl, and a replacement can be made.
It's a wonderful thing to recognize fundamental science that has led to discoveries that I think will have a real impact in the future, and also to recognize the large body of work that has gone into this technology development.
What exactly is CRISPR-Cas9? It was not originally intended as a genetic editing tool. It was actually a defense system found naturally in some bacteria and archaea. Pictured here is a phage invading a bacterium. More often than not, a phage will insert its DNA into the host, leading to the host destruction. But if the cell is lucky enough to survive the attack, it will incorporate part of the attacker's DNA into the CRISPR array located within its own genome. In a sense, it remembers what the attacker looked like. The bacterium then transcribes cRNAs from this CRISPR array, including sequences that can recognize viral attackers that it has seen before, and sequences that can bind to another critical RNA called tracer RNA. These two RNAs bound together can act as a GPS for an enzyme called Cas9. When phage DNA reappears, it guides the Cas9 protein to the invader and unwinds the invading DNA to check its identity. If there is a match to the cRNA, Cas9 will activate two of its molecular scissors and cut both strands of the invading DNA, thereby incapacitating it. Microbiologist Emmanuel Charpentier was the first to identify that 2 RNAs were needed to operate the system. She realized that by customizing the cRNA, we can control this tool to search for any DNA sequence and not just viral DNA.
Bioinformatically, 2 RNAs that were expressed in S biogenes. So one is a tracer RNA and the other one is a CRISPR RNA of the CRISPR-Cas9 system. And along the way, we became interested in understanding how the system was working. And we found out really that this enzyme is working with 2 RNAs and can be programmed as a technology to perform genome engineering.
UC Berkeley professor Jennifer Doudna, in collaboration with Charpentier, combined the 2 RNAs into a single guide RNA, further simplifying this process into a technique that any laboratory can use.
The editing, in principle, could be done outside the body by removing stem cells of the blood from a patient, correcting them outside the body, and then putting them back in only after those edits have been verified to be correct.
The use of this technology is not limited to the test tube. It can also be applied to the nucleus of living organisms. Now, what was once a bacterial defense system can be used to manipulate DNA with ease. A pre-programmed single-guide RNA can guide Cas9 to any gene within the genome, where it can then make a cut. When the cell attempts to fix the cut DNA, as it naturally does, researchers can slip in a specific piece of DNA and incorporate it into the genome. This method is far cheaper and far more accurate than any gene editing technology known before.
So the system is programmable in an easier fashion, and it's also less costly, and it's also more versatile. This is important to say, in the sense that CRISPR-Cas9 allows not only to act directly on the DNA, but also to change the expression of the DNA.
It was around the same time that Feng Zhang of MIT was performing his own modifications to the Cas9 system. He was the first to demonstrate that the system could be used to successfully perform genomic editing to the eukaryotic cells, mice, and even humans, taking CRISPR-Cas9 technology to a new level.
We have also applied it to generate mouse and cellular models of autism spectrum disorder so that we can understand what are the mutations that play a causal role in the disease, and then what is the mechanism that this mutation sort of functions in the body. We've done this now in mouse models and human stem cells. And also we try to develop the system so that we can test whether or not it can have therapeutic potential.
CRISPR-Cas9 is now being used to find cures to genetic diseases. It could potentially be used to treat sickle cell anemia, fix mutated genes that drive cancer development, or cure AIDS by eliminating HIV from human cells. CRISPR-Cas9 could also be a game changer in agriculture, offering a more controllable way than previous GMO methods to yield more robust, more bountiful crops.
There are also ways to use this to generate improved plants. We can use it to screen for new traits that confer either viral resistance or drought resistance, make crops more robust, and be cultivatable even in very harsh conditions.
To cut and paste genes in plants is one thing, but to edit mammalian or even human embryos is an entirely different consideration, not something to take lightly. Modifying the genetic code of an embryo means a permanent change that will be passed down to future generations, forever altering the course of human evolution. This may sound like a good sci-fi story, but in reality, it enters ethically uncharted territory.
Somebody that wanted to have a child that was very tall or had dark hair or something like that. Right now, for the most part, we don't know the genetic basis for those kinds of traits, and so therefore we can't really engineer someone even if we wanted to do that. But I think what's happening is that over time, and certainly with technologies like Cas9, we meaning scientists in general are very enabled in terms of our abilities to figure out the functions of genes, including in humans. And so as we learn more, there will be increasing, I think, opportunities and, frankly, demand for changes to be made to the human germline.
Genetic changes like this are still in the theoretical stage, but already CRISPR-Cas9 is being used in human stem cell engineering, disease mechanism research, drug screening, and much more. These scientists have propelled CRISPR-Cas9 into a transformative technology simple enough for any researcher to use. It is also remarkably accurate with a specificity over 80%.
德纳跟夏彭提尔的工作的确是把那个 Cas9 那个 enzyme inside out， 他可以把它研究得非常好，让他知道可以要用哪些 RNA 可以让它的活性更强，然后怎么样变成一个 single guided RNA， 他是从它的那个结构的方向慢慢做出来的。张峰这就想的不一样。张峰虽然也会做这些，但是他想到说，我要知道说怎么样放进哺乳类，怎么样在哺乳类能够做过，所以他比较跟细胞直接有关，跟这个工程、生医工程有关。所以这是他们其实是相当不同的，一个是从比较是生医工程的角度，比较是有应用性。那另外一个是比较是，从刚开始是从比较基础的科学开始哈，可是后来当然也都会想到应用。
Emmanuel Charpentier, Jennifer Doudna, and Feng Zhang. Naturally, these scientists thought of the real-world implications of the technology, but each from the perspective of his or her own expertise, and each with a unique vision. Through countless hours in the lab working on this new and unknown mechanism, they brought this tantalizing technology to the light of day. With CRISPR-Cas9, they expanded the boundaries of genetic engineering and discovered whole new frontiers of possibility. Throughout history, scientists have changed the world and the human race through their discoveries born of passion and curiosity. In the case of French microbiologist Emmanuelle Charpentier, we see that same curiosity and passion at the heart of her research. The pursuit of truth has always been central to her life, and she even once considered being a detective. But work with experience at a children's research hospital convinced her that research was her way to help humanity.
I've always been interested in art because I started very early on at 5 years old with ballet dance, and at 6-7 years old with classical piano. So this has been part of my life for a number of years.
Impeccably dressed with swift, lively eyes and an easy precision with words, she embodies the air of European charm and wit. In addition to music and dance, Charpentier enjoys learning about the cultures of different countries. Art, paintings, and relics are of great interest to her. She might very well have become a classical musician or perhaps a world-renowned dancer, but Charpentier knew that she was fit for a different calling. Music and art give her peace of mind and a creative release, but it is in the laboratory where she feels whole.
So because I was always curious, so I thought that going in this direction would allow to somehow fulfill my curiosity. I think this is the main drive. And so I think the fact to be able to really focus on certain questions and be persistent to really answer those questions, it's really something that I think fits well also my personality, and something also that I can fulfill being a scientist in an academic environment.
Charpentier studied biochemistry and microbiology at the University of Pierre and Marie Curie in Paris, then continued her work at Rockefeller University, New York University, and St. Jude Children's Research Hospital in the U.S. 6 years later, she returned to Europe to start a lab at the University of Vienna. In 2009, when she was switching institutions to her new position at Umeå University in Sweden, that she experienced what can only be called a moment of eureka.
And essentially to really connect this tracer RNA molecule to the CRISPR-Cas9 system. So the research was conceived in a way during my move to Umeå University.
While Charpentier was at Umeå University, she focused on studying the CRISPR-Cas9 system, working day and night in the lab, eager to unveil the mystery of this new mechanism. In the evening, as the lights clicked off in other laboratories, Charpentier's laboratory remained illuminated as she continued enthusiastically into the night.
When I had a late experiment and I would end at 1 or 2 o'clock in the morning, and I would meet her on the corridor, and I always thought of myself as the hardest working person in the department, and in fact Emmanuelle was always working at night, and she's a really extremely hard worker, and she was essentially all the time in the lab, and this is how we met the first time.
People may think such long hours excruciating, but to Charpentier, being in the lab, focusing on science and digging into life's mysteries actually gives her a sense of freedom. In 2015, Charpentier was recruited to become the director of the Max Planck Institute for Infection Biology. She moved to Berlin. Bringing cutting-edge research on CRISPR-Cas9 to Germany at such a critical time has opened up exciting new possibilities for many German scientists.
And I think to have Emmanuelle in Berlin is really the best, because we may be the first who can work with her together, who can participate on her knowledge, and who can develop the system to another level where we can really bring it in patients' care.
Emmanuelle is a wonderful young scientist, very ambitious, very nice to collaborate with, very strategically thinking. So yes, I think we are all enthusiastic and we are very, very happy to have her here.
After coming back from lengthy postdoc training in the U.S., Charpentier seldom returned to her homeland of France. Instead, she followed the science from one country to the next, always in search of the best place to continue her research.
She lived in 5 different countries. She joined 9 different institutions, if I'm not mistaken. And all this, I think, shows you how energetic, how driven she is, and how passionate she is about science.
You know, in a very short time became very famous. She's very approachable. She spends time with people. She is, you know, a very wonderful colleague and always approachable and nice to talk to, very open. So yes, she will be an inspiration for the young people, but also for the older guys like me.
Curious and passionate by nature, Charpentier loves meeting and interacting with researchers from different fields. When it comes to talking science, the pure joy of sharing and learning new ideas is apparent in her demeanor. Colleagues often mention it as one of her most unique qualities.
I learned while I was still in New York during the summer that Emmanuel, Dr. Charpentier, was also recruited to join the Max Planck Institute here in Berlin. And so I immediately looked up her contact information and sent her an email. On a Sunday afternoon, I was still in New York. And I didn't expect an answer immediately. After all, we hadn't known each other before, and I just introduced myself via email. But she made the effort to call me back right away, the same day. As a matter of fact, it was already midnight in Germany, and so I was kind of blown away. I mean, she was very clearly interested and interesting, and I was looking forward to establish contact and discussion with her.
Charpentier's innovative thinking may come hand in hand with her interest in different cultures, different peoples. It was during one such international exchange that she met Jennifer A. Doudna, with whom she would develop the bacterial defense system into a powerful new gene editing tool. Born in Hawaii, Jennifer Doudna is a professor of chemistry and of molecular and cell biology at the University of California, Berkeley. She is also a member of the U.S. National Academy of Sciences. In collaboration with Charpentier, Doudna is credited with fusing CRRNA and tracer RNA into a single guide RNA, resulting in a rapid, powerful tool for genome targeting.
We met at a meeting in Puerto Rico in 2011. And when we got together at the conference, we hit it off personally, and we realized that our labs had different expertise.
Great discoveries are often highly complex and require the knowledge and cooperation of different disciplines. One lab can only do so much. When several labs cooperate, however, the results can be exponential.
Her lab works on medically important microbes, so pathogens, bacteria that infect people and lead to disease. And our lab was working on the fundamental molecules that are involved in CRISPR systems and other kinds of gene regulating pathways that use RNA. Mm-hmm. And so we recognized that there was a nice opportunity to work together to figure out the function of the Cas9 gene.
Bright-eyed, affable, passionate about science, Jennifer Doudna lights up with the warmth of the Californian sun whenever she starts talking about science. Today, she and her husband, Jamie Kate, take one of their strolls through campus. You may wonder, how does a researcher so accomplished find balance between work and family? The answer is simple. Make sure your husband is in the same department as you. The Campanile Tower stands high over them, its bells tolling the hour as they discuss data from the lab, new ideas, or what to eat for dinner. It silently watches as these kindred spirits walk hand in hand down the path of love and science.
So I actually, after finishing graduate school, I moved to California while she was at Yale, and I was working in Professor Harry Noller's lab on how proteins are made in cells. And so we were on opposite sides of the country, but we still kept in touch, and then she started dating, and that's how we got together. We actually got together while we were on opposite sides of the country. She's very passionate about science and the process of discovery and invention, and I think that's what really drives her to come in every day. And she's very social and loves talking with students and postdocs and other scientists about project ideas, and so I think that carries over every day.
Kate said that if he had one word to describe his wife, it would be passionate. He says that his wife is more determined and more willing to embrace challenges than anyone he has ever met. Perhaps these qualities are what brought them together.
After what they're most passionate about, not necessarily think about how it might be applied down the road, because that's not always easy to tell. And so I think all along she's been very driven to go after questions that she found the most interesting.
Developing a new technology requires time and energy and often presents obstacles at every corner. But Doudna constantly reminds herself an obstacle just tells you that you're not there yet. She trusts her instincts and believes that if you're tenacious enough in getting to the bottom of a problem, you can overcome any difficulty along the way.
Don't let anybody discourage you from what you think is important and interesting to study. I can't even remember how many times people in my career and in my life have told me, "Oh, don't work on that. It's not interesting," or "That's too hard. It'll never work." And I've always just I listen to that, but I don't always follow that advice. If I think something is really worth doing and really important to try, I try to go after it, and I try to convince other people to work with me to do it. It doesn't mean I'm always successful. In fact, most of the time, those kinds of things don't pan out. But every now and then they do, And when they do, it's an exciting thing. And I realize, looking back, that if I hadn't taken those risks in my career, that I would have never gotten to where I am now.
What lies behind Doudna's passion is a fundamental curiosity of how things work. Every once in a while, in the process of answering one question, a science finds an answer deeper and more vast than they ever imagined. Such was the case of CRISPR-Cas9. What started out as studying the function of bacterial defense system turned out to be a major breakthrough in biology.
I think that, if you look at her career, she's always been successful in whatever project that she's picked up. And I think it's, again, because she just has a knack for thinking about what's the major question or the missing knowledge in a given field. And so she's willing to go off into some new area because she knows that that's where there's the biggest gap in our knowledge. And I think that there are other scientists who have that ability as well. And whichever field she's gone into, she's been able to do that.
Doudna's husband compares her to a curious butterfly, fluttering from flower to flower, investigating, exploring the world. And it may be her butterfly-like ease, her passion for discussing ideas, that makes her so well-liked among colleagues and also people from different fields.
So my son often comes to lab here after school, and at the end of the day, all three of us will go home, and Andrew and I will be making our way to the elevator, and sometimes it's hard to get Jennifer out of the building because she's so social. And I have this phrase for it. She has social viscosity, high social viscosity. So if she bumps into somebody in the hallway, start talking about ideas. So I think that kind of passion for ideas is really something that's helped her succeed.
When she visited Taipei in 2016 to receive the Tangprize, she had the opportunity to interact with high school and college students. Students filled the auditorium to listen to her experiences and ask questions. It was clear from the smile in her eyes that she enjoys interacting with the youth as well.
Something very interesting.
If we can really create an ideal baby, like 100%, they are smart, they are brilliant, they are pretty. So is there any further limitation about this research?
The kind of thing that you just mentioned is probably still, a couple of decades away, really. But I guess my point in discussing it really is just to point out that it's coming, that time is coming, and it's not a fantasy anymore, right? It's not a science fiction. It will happen.
If gene editing technology can ultimately be used in human embryos, future human beings will be able to free themselves from genetic disease, but also it gives humans the choice to say goodbye to imperfections. Dowden encourages scientists to slow down, get out of the lab, and talk about the ethical issues involved. While CRISPR-Cas9 is still at an early stage of development, the technology is already powerful beyond anticipation. Its effects on agriculture, the environment, and possibly the human germline are changing the course of evolution. Dowden believes that the time is now. Scientists must rally people with different specializations, not just other scientists, people like teachers, artists, directors, and writers, and spread knowledge to the public so that society can make informed decisions.
I think it's through having scientific meetings that are open to the public, and already some of that is happening. But I also think that it's through working with artists, working with musicians, working with writers, working with journalists, working with movie makers. These are all people whose jobs, really, in many ways, are to create either whether it's artwork or writing or videos and documentaries and things like that communicate ideas to a broader public. And so I think by having scientists interacting with people that are not scientists but whose jobs are really involved reaching out to the public on different topics, that we can really engage people on a much more broader level.
Passion attracts passion. Charpentier and Dowden value each other as like-minded people, even though they come from different backgrounds. From deciphering the long-standing mystery of CRISPR-Cas9 to making a significant upgrade to our genetic toolbox, the two have developed a close rapport.
I think she's very stylish. She's from Paris. She is very passionate. I was drawn immediately to her intensity as a scientist. I think she has a great sense of what's important in science and what the big questions are in science. I think she really recognized that the power of the genetics and cell biological experiments that her lab was doing could be extended by working together with a lab like mine that does biochemical and structural biology work. So, and she has a wonderful sense of humor, so we've had a lot of good laughter together.
Me, I like to collaborate. I think this is the same thing for Jennifer Doudna. I think she's also a scientist who collaborates very easily. And I think we have a little bit maybe the same view and that's why we connected very fast.
The list of contributors to the CRISPR-Cas9 platform doesn't end here. We now move to the eastern seaboard of the United States, where Feng Zhang and his team are hard at work.
We have about 20 graduate students and postdoctoral researchers, and we work on a combination of developing CRISPR-Cas9 or CRISPR CPF1 genome editing technologies to be able to study biology, but also trying to turn it into a therapeutic ready molecule.
Today begins like every day. Zhang dons a lab coat and gets to work. At age 34, Zhang was already an indispensable investigator at MIT. His team's goal was to make CRISPR-Cas9 work in human and mouse cells. Not only were they the first to successfully target genes in mammalian cells, they also showed that they could edit several genes at once, opening the doors for discovery and development around the world.
We can use it both in research to really advance our understanding of disease, so that drugs are really developed based on a rational understanding of the mechanism, so that we'll have a much higher likelihood of succeeding. In addition to that, you can also imagine developing real therapeutics to treat a number of different diseases.
Zhang was born in China and immigrated to the U.S. at the age of 11. Most teenagers spend time in front of the TV, watching movies or playing video games. For Zhang, one such movie would become a turning point in his life. In one Saturday enrichment class in molecular biology, Zhang saw the 1993 blockbuster Jurassic Park. People have praised the movie for its special effects, its memorable ending, and even its musical score. But what really hit Zhang was the possibility of engineering life.
I had no idea what molecular biology is. My impression of biology was memorizing different types of trees, dissecting small, really smelly sort of paraformaldehyde preserved frogs and labeling anatomical parts. That was not so interesting to me. But then when I went to this class and they showed us Jurassic Park and taught us some of the advances in molecular biology, I got really captivated.
In Jurassic Park, scientists use molecular cloning to bring back dinosaurs. While most people are on the edge of their seats with the terrifying realistic effects, 13-year-old Zhang viewed it as a documentary. It taught him basic principles governing life, including how DNA is translated into proteins and how it is possible to engineer this genetic code to reprogram biological systems and, ultimately, change reality.
I got really captivated because all of a sudden, biology is no longer just about memorizing things. There are fundamental principles, like DNA is formed by just 4 DNA bases, and they encode amino acids. So there's something there's a fundamental principle that kind of governs how this whole biological system, how they work, and I like that, and I thought if I can learn to understand these principles better, maybe I can use it to engineer useful things.
Every scientist was once a teenager, and getting into trouble is what teenagers do. His longtime friend Sid recalls one of those memorable moments.
Feng and I have known each other for 17, 18 years now. We first met at the Boston Logan Airport. We were both waiting for our luggage after flights from the Midwest, and I saw this kid with this science t-shirt, and so I walked over to him because I thought that he was also going to the science camp. So I asked him and he said yes, and ever since then we've been best friends. One funny story is we were actually lab partners back in college in organic chemistry our sophomore year. But in this organic chemistry lab he forgot to include one of the reagents before we actually started heating the mixture, and then afterwards thought, okay, well, maybe I should just include it now. What would actually happen? And then there was this explosion and all of the experiment had sort of fallen apart. And I just find it interesting because now he's a well-known and very successful scientist, but every scientist has some mishaps along the way that they learn from, and that probably was one for him.
Zhang received his bachelor's degree from Harvard University in chemistry and physics. Then, after receiving his doctorate in chemistry and bioengineering from Stanford, he joined the Broad Institute of Harvard and MIT, where he became the youngest ever core member. From dawn to dusk, Zhang is in the lab learning new things, testing new ideas. He believes that in order to break new ground in science, different ideas, different people, different perspectives must come together and interact. His students and postdocs are a big source of inspiration.
For me, when I was rotating in the lab, I just loved being in the lab, and so I would end up being here very late until like 11, midnight, and it was funny because not that many people would be around then, but Feng happened to be around like every night that summer, and he would, like, my rotation desk was over by the side and it was next to his bench, so he would be like doing experiments next to me like 11 p.m. I was like, I've never seen a professor, actually doing experiments, staying in lab late, like along with his students.
There's something about Zhang's personality that makes him genuinely likable. It could be his gentle demeanor or sense of humor, but it may also be his sincerity and humility. His clear logic and foresight have led to one success after another, but Zhang never takes excessive pride in his accomplishments. Zhang's eyes shine with excitement when looking over some promising new data or when mulling over an intriguing new idea. In short, his enthusiasm for research is contagious. Students love being around him. They work on the same bench, live the same lifestyle, and coincidentally, might even be the same age.
I remember that we were working on a paper and it was my first paper, and I was super excited about it, and Feng actually let me like submit it because he knew it was like such a big deal. And then we pulled an all-nighter. We actually pulled a double all-nighter, which I'd never done in undergrad, and all of the grad students we all like passed out and had to recover, but he had a meeting at like 7 a.m. and so he just had to go to that, and so he was really just tireless, and he just does this great thing where he just engages with you, and you get a really strong personal connection. It's a crazy experience, and we got to like talk about science, and he took us out to dinner, and it was ridiculous like that this professor who was doing so much in lab also had so much time to kind of dedicate to his students.
Well, he is definitely one very, very well read, and he knows a bit about everything, and it's amazing to learn from someone who has such great insight on almost every aspect of biology, and that's how he has an environment.
Even though people in the lab look up to him, Zhang never places himself above others. Whenever he has the chance, he joins his students for lunch. They run the gamut from science to life, because to Zhang, they are not only his students and colleagues, but also his friends.
Feng's general presence all the time is the reason that the lab is so successful. Feng's here late at night with us. We go out to dinner and we talk about science, and Feng's always there. So there's always sort of like a general like moving forward, and Feng is the driver. So yeah, of course that leads to success.
If Feng Zhang's laboratory is a ship, then Zhang is the captain or the navigator, taking them to uncharted lands. But no matter how uncertain the waters, the passengers feel secure because they have faith in their captain, who pushes forward at an astonishing pace. But even ships need to make brief stops. Research is critical to Zhang's career, but nothing is more important than a friend in need.
Most recently, when my grandfather passed away, I think he has a lot of stuff going on, as evidenced by the reason that you guys are here. But the moment I mentioned it to him, he and his wife dropped everything, said, "What are you doing right now? Where are you? We want to come see you." And they spent a couple of hours with me just talking through what had happened, how I felt, how my family was doing. And it was just comforting to be there with a friend who could be there for me in a moment that I needed it.
Some people attribute Zhang's early success to genius or an obsession with science. But to Professor Guoping Feng at MIT, someone whom Zhang respects as a mentor, modesty and kindness are what brought Zhang such great success. He remembers one such instance that moved him deeply.
When we also have collaborations internationally, work together, and last a year ago, we actually started a collaboration with the Chinese Academy of Sciences in Shenzhen Institute of Advanced Technology, and we need him to go there to defend our proposals and to get funding. He would just fly there just for that half day, and all the way to Shenzhen in China, and give the talks and help to prepare things, then come back because he has a very busy schedule like everyone else, probably busier than most of us. And he would just go there, flying for a total of about 36 hours just for that half day over there to help us to get that collaboration going. Wow. So that's kind of warm, supportive personality he had.
With a sincere heart, humble attitude, and perseverance, Zhang has won wide acclaim. He has won the admiration of his mentor, inspired his students, and kindled the curiosity for science in the people around him. These three pioneers of the CRISPR-Cas9 technology have worked tirelessly to unlock the potential of this gene editing system. In the small, quiet hours of night, research can seem monotonous, frustrating, unending. But the next great discovery may be just one more minute away. CRISPR-Cas9 has been there, waiting invisible to the naked eye, in something as inconspicuous as bacteria. Like the spark of curiosity in these three scientists, CRISPR-Cas9 started small but will change history and humanity in colossal ways.
Just a few weeks ago, University of Pennsylvania, their RAC committee approved a strategy of using the CRISPR system to treat blood cancers. And also, I think even just a couple of weeks ago, clinicians and scientists in China announced that they were going to develop a CRISPR-based therapeutics for engineering immune cells to be able to attack cancers in the body. And so these are very exciting areas that I think we'll see a lot of progress in.
In fact, CRISPR-Cas9 gene editing technology is entering a new stage of human clinical trials.
The fact is that the CRISPR-Cas9 technology is really meant to be used in the laboratory for research and development purposes in any field of biology one can think of, whether it's a medical-oriented field or agriculture-oriented field or biotechnology-oriented field. Now, surely there is a hope, and this is an effort that is carried on by a number of scientists in the academic setting, in the clinical setting, and also in the biotechnology setting, is the fact that the technology could be really developed as a direct technology to really treat human genetic disorders. Now, you have some risks with regard to wrong usages of the technology, but I really rely actually on the fact that you have anyway a number of biosafety and ethical regulations which the scientists are following up.
More attention should be put on the ethics surrounding CRISPR-Cas9. In a very short time, it has already changed the way we do science and the way we plan to treat diseases. It even has the power to alter fate itself, so we must be careful of the ethical implications.
If this technology is employed in embryos or germ cells, eggs or sperm, the changes that are induced in the DNA become inheritable, so they can be transmitted to future generations. Mm-hmm. Something that doesn't happen when we do somatic cell editing, which would be used in one patient to cure one disease, mm-hmm. and that's it. Right. And so, the opportunities with human embryo editing are enticing in a way, because one could imagine correcting a genetic disorder that would then never be transmitted to future generations. But it also means that scientists now, in principle, could make changes to the evolution of human beings, right? Because we now sort of have control over our own genetic inheritance. And so that's something that I think is just it's clearly very powerful technology, and something that requires careful thought before forging ahead.
Ever since these scientists published their discoveries in 2012 and 2013, more than 4,300 articles have been published on CRISPR biology. Not only can the system be used to precisely edit the genome in virtually every kind of laboratory model, other technologies derived from the CRISPR system are also in development. But Stephen Lean, an investigator working on CRISPR at Academia Sinica, believes that the true potential for this system is in gene therapy.
So, as we know, we have very good chemistry and drug screening system. So we can design good drugs, but those patients, they have to take drugs throughout their life. So why not, with the advance of programmable nucleases, perhaps it's a better option to go back to the DNA blueprint and then fix the problem from the source. Yeah, so that way patients, they can be cured. Yeah, their diseases don't have to be controlled throughout their life.
At the same time, around the world, scientists are helping to improve the CRISPR-Cas9 system so that it may one day be used in the human body to cure the incurable and benefit all humankind.
人类在疾病上，在农业上，在工业上，都会有非常非常大的功用哈，因为它可以，比如说在不是人的话，你可以改造基因，那些改造基因只要现在已经在做了，对不对？现在即使不用 CRISPR 已经在做了，也可以帮助增加绿能，慢慢看怎么样非常严谨的定些规则啊，或者严谨的来考虑这件事情，所以我相信将来 我再讲，就是我对 scientific community 是非常乐观，我觉得他们会很小心，所以这个 CRISPR-Cas9 将来是 potential 非常非常大，所以我并没有太多的，我个人没有太多的疑虑。
CRISPR-Cas9 promises to change people's lives. From the blooming of flowers to the treatment of illnesses, there has never been a gene editing technology with such wide impact. The 2016 Tangprize in Biopharmaceutical Science awards these 3 young scientists for their contributions to biomedical research and for helping scientists to make significant leaps towards understanding human diseases, on top of just understanding genetic defects. CRISPR-Cas9 provides humanity with a technique to actually correct them. Before CRISPR came along, it is almost as if scientists had their hands tied, dreaming that someday they could edit living organisms precisely and effectively. Thanks to this year's Tangprize awardees, that someday is today.